Hello students. Today we are going to discuss Unit 3 Life History of Cultivated Fish and Prawns from BSc Second Year, Semester 5. In this chapter, we are going to learn life history of rohu, life history of other cultivable fishes, life history of mussels, life history of cultivable prawns. So it is important to know about different life stages of fishes and prawns as it is intended for fish farm culturing. Knowledge about spawn, hatching, fry, fingerlings, and adult is necessary in fish farming, while prawns show larval forms, which develops into zoea, mysis, post-larval stage, and finally adult. If you look at the life history of rohu, it attains maturity towards the end of the second year of life. It spawns only once in a year, and the spawning duration is short. The spawning season of rohu generally coincides with the southwest monsoon. Fertilized egg of rohu is round, transparent, non-adhesive, and red in color. The yolk spear contains oil globule. Fully fertilized eggs are 5 mm in diameter. The newly hatched larva measures 3.5 to 4.5 millimeters in length. It has a transparent, laterally compressed body and is characterized by the presence of gill slits, fin, and median fin fold. The yolk is gradually tapering with a prominent anterior portion and elongated and narrow posterior blunt end, having slight pinkish hue on its dorsal side. The hatching has faint yellow, fish brown eyes and having slight pinkish hue on its dorsal part. The hatching shows irregular movements occasionally or remains laterally at the bottom. This is a picture that shows the life cycle. So 30 minutes after fertilization, the cell division takes place. You can see the cleavage here, and it changes into a spear-like stage, which is having around 1000 cells. Then the next stage is called as shield stage. Then the gastrulation takes place. This is the gastrula, and it is called as epiboly. Then epiboly changes into the next process, which is called as organogenesis. The organ starts forming, and then it's called hatching stage. Now after hatching, the free swimming larva looks like this. And then the larva grows and changes into an adult fish. Spawn just after absorption of yolk measures 6.5 mm to 7.5 mm. Rudiments of dorsal, anal, and pelvic fins appear on the 30th day, barbels on the 21st day, and liver on the day after hatching. The air bladder originates 2 inches per 2 days after hatching. Now let us see the characteristics of the different growth stages of rohu after hatching. Now, how does it look after 24 hours of hatching? Average size of larva is 5.5 millimeters. A few black chromatophores are seen on the head above the eyes. Embryo is pale yellow in color. Anus is distinct. Auditory concretions clear. Yolk sac ends sharply distally to a point. Striations are seen on the caudal. Pectorals are without rays. Notochord turned upwards only at the end. Now, this is a general life cycle of a fish. Fertilized eggs gets converted into larvae. Then the larvae changes into fry. Then it converts into fingerling. Then the next stage is called as juvenile. Juvenile changes into adult fish, and then it attains the spawning adult, the spawning stage. Then again it starts laying eggs. This is how the eggs of a fish look like. Now let us see what happens and how does it look after 36 hours of hatching. Average size is about 5.9 mm. Lower lip is clear. Pectoral fin is prominent. Anterior profile of yolk sac is more or less straight. A few black chromatophores are on dorsal margin of the yolk sac throughout its length. A few on the dorsal fin. No chromatophores on caudal region. Dorsal and ventral sides of embryo are pale yellow in contrast to the deep yellow color of the caudal region posterior to the anus. Notochord slightly upturned at the tip. Mouth appears as a slit. After 48 hours of hatching, average size is 6.2 mm. Yolk sac is convex anteriorly. Embryo is yellow in color. Air bladder is distinct. Pectoral fin is prominent. A few black chromatophores are on the head, behind the eyes, and in the auditory region. No chromatophores on the ventral side of the air bladder. Black chromatophores are in a row from the area posterior to the auditory concretions up to the base of the caudal fin. Large black chromatophores are on head prominent. Ventral embryonic fin originates in advance of the dorsal fin fold. Head is dark and body is faint yellow in color. After 72 hours of hatching, average size is 7 mm and looks pale yellow in color. Black chromatophores are on the head with few in between the eyes. Dorsal profile of embryo is bright till the anal region. Caudal region above the notochord is yellow in color. So the developmental stages of Rohu after 3 days of hatching are as follows. Stretching from abdominal region up to anus, dorsal embryonic fin fold is visible as far forward as opposite to the anus. Two black chromatophore clusters are at the caudal peduncle and two crescent-shaped patches on caudal fin. Caudal region is with 22 branched rays. Caudal fin less deeply forked, pelvic fin with 2 to 3 rays, dorsal with 2 to 11 rays. Fourth day after hatching. It is 7.6 mm in length. Yolk sac is completely absorbed on the fourth day after hatching. Lip margins are gently fimbriated. Distinct black chromatophores are seen behind the eyes on the head region. Opercular outline is distinct. Notochord bent at the tip. Crescent-shaped semicircular area below notochord in the caudal region formed of black chromatophores in rows. The area of the semicircle is well defined. Dorsal and ventral fin folds are persistent. Commencement of caudal rays are seen. Sixth day after hatching. It is 10 to 10.5 mm in length. Lower jaw is bigger and upturned. Gape of the mouth is 1.7 mm and eye is 1 mm. Nostrils are prominent. Dorsal fin is with 9 rays. Anal with faint rays. Pelvic fin bud seen. Air bladder is divided into two. The anterior part, globular and posterior in the form of elongated triangle. Dorsal side of the body is yellow. Chromatophores form two crescents on the two lobes of the caudal fin, separated by a small colorless area. Caudal rays are 22. Seventh day after hatching. Now it is 11 mm in length. Lips are thick. Notochord is sharply bending upward. Ventral embryonic fin fold. Then, eighth day after hatching. It is 12.5 mm in length. Dorsal fin with 14 rays with sparsely distributed yellow pigment near base of the rays. Annals are with 7 rays. Ventral embryonic fold is ending in the anal region. Caudal is with 22 rays. Dark crescentic areas at the base of the caudal fin and a few black chromatophores on the embryonic fold connecting the caudal fin with the anal fin. Pelvic fin is with fin rays. Body is golden yellow. Dorsal half is more predominant. Prominent black chromatophores are on the head. Black chromatophores scattered all over the body, exhibiting no pattern. 12th day after hatching. It is 19 mm in length now. Maxillary barbels are present. Body is golden in color on the dorsal side and dirty yellow ventral side. Dorsal fin with 3 to 13 rays. Anal fin, it has 7 rays. Except for the anterior one, the rest are branched. Ventral fin is with 7 rays, except two anterior lines. The rest are branched. Caudal fin rays are 34. A broad triangular black band on the caudal peduncle across its entire length with the apex facing the head. Next, 15th day after hatching. It is 23 mm in length now. Anal fin with 2 to 5 rays. Caudal rays 36. Basal 2 by 3 of the dorsal fin pigmented. Black chromatophore band in caudal region, posteriorly prolonged from the center, passing through the origin of caudal rays and dividing the fin into two. Next is 18th day after hatching. Now the size has increased from 23 mm to 25 mm in length. Body is dirty yellow in color above lateral line and yellowish white below that. Scales present but not clear near the caudal peduncle and in the abdominal region. Black chromatophores are on the anterior margin of the dorsal fin in the form of dotted line. Ventral fin is with 9 rays, 32 caudal rays, two prominent grayish crescents, one on each half of the caudal fin lobes, behind the dark triangular areas on the caudal peduncle. Now, look at the size after 20th day after hatching. Now the size has increased to 26 mm. Now the body from dirty yellow color it changed into golden color. Now scales are prominent and fully they're covering the body. Two dark bands, one above and one below the lateral line, are along the body up to the caudal region. Dorsal fin is with 3 to 13 ray. Caudal rays are 34. Rays covered with orange pigment dots. A black band along the entire width of the caudal peduncle, concave posteriorly, and are irregular in shape along the anterior margin. Behind this band, two faint crescentic areas separated by two colorless streaks. Then 25th day after hatching. It is 30 mm in length. Body is golden colored. with coppery reflection from operculum. Barbels are prominent. Upper margin of eye is faintly orange. Dark band in caudal peduncle is quite prominent. Upper lobe of caudal fin larger and pointed compared to somewhat round edge of smaller lower lobe. Now let us see the life history of other cultivable fishes. Of the exotic food fishes, these are more important from the point of fish culture. The one is common carp, Cyprinus carpio, silver carp, Hypothanictis mollitrix, grass carp, Tinopharyngodon idilla, tilapia, Orchochromis mozambicus. If you see the first one, common carp, that is Cyprinus carpio, the fertilized eggs of common carp are small, spherical, demersal, and adhesive, measuring from 1 to 2 mm. The pervitline space surrounding the egg is narrow. The perivitline space. Yolk is usually yellow to light brown in color. Embryos hatch out within 2 days to 7 days, depending on the water temperature. The newly hatched larva is 4 to 5.6 mm in length and has a prominent yolk mass. The newly hatched larva adheres to the leaves of aquatic plants by means of cement glands and remains in these conditions until the yolk is partially absorbed. The yolk is absorbed within 2 to 6 days depending on water temperature, after which the post larvae measuring 6 to 7 mm start normal movement and commence taking nourishment from the environment. If suitable food is available, the growth is rapid and fins get differentiated. Within 12 to 15 days of yolk absorption, all fins develop the full complement of race. Now, this is how the common carp looks, and it grows very, very big in size. Next, let us discuss about silver carp. So, its zoological name is Hypothalmictis mollitrix. The freely oozing viable ova of silver carp are pale bluish in color. Fertilized eggs are demersal and round and ashy gray in color. Within 5 minutes of fertilization, eggs start swelling, and during the next half an hour, the process of water hardening is completed. The full swollen fertilized eggs measure 4.2 to 4.76 mm in diameter. The length of the newly hatched larva of silver carp is 5 to 5.5 mm. The length of yolk sac is 3.33 mm. Height of the body is 1.25 mm. Height of the yolk sac is 1.09 mm. With 42 to 44 in number of myotomes, of which 14 to 16 are located behind the anal level. Yolk sac are fully absorbed by the end of the second day of hatching, and thereafter spawn start feeding from the environment. They measure 7.42 to 7.53 mm in total length and 1.21 mm in height of the body. This is the picture of silver carp. The name only suggests the body is silverish in color. It's so shiny. The next one is grass carp. This fish lays eggs in stages. Eggs are pelagic and undergo development as they float downstream. Ovarian eggs are of yellow and deep golden brown in color. Fertilized eggs get fully swollen in half an hour and measure about 4.58 mm in diameter. Newly hatched larvae measure 4.5 to 6 mm in length. Length of yolk sac is 2.84 mm. Maximum height of the body is 2.90 mm. And maximum height of yolk sac is 0.78 mm. Yolk mass is conspicuous and of a pale brown color. Of the 45 myotomes, 14 are posterior to 37 at the anal level. 2-day-old post larva, just after absorption of yolk sac, is 6.98 mm in total length and 0.99 mm in maximum height of the body. Let us see the picture of the grass carp. Now let's see the life history of morels. The fertilized egg is round, non-adhesive, buoyant, free and straw yellow in color. The yolk is capped by a distinct blastodisc and contains an oil globule liable to disintegration to smaller 5 to 6 globules, unequal in size. Perivitelline space is of medium width. Diameter of the eggs is 1.2 mm and that of the yolk is 0.9 mm. 10 hours after fertilization, anteroposterior axis is distinguishable, cephalic portion being broader, forebrain region discernible, invasion of the yolk is complete. 14 hours after fertilization, the embryo with 6 somites is embedded all over its length in the yolk mass. Optic cups are clear. Height of the embryo is 1 mm. 16 hours after fertilization, mesodermal 9 somatids are present. Notochord is more discrete and the fore, mid and hindbrain regions are also defined. Cephalic portion is broadened. 17 hours after fertilization, the somites range from 13 to 15 in number. Ectodermal thickening to form lens of the eye is indicated. Tip of the tail is from the yolk at the last somite. Embryonic fin fold appears. Copter's vesicle is visible. Cephalic region is depressed. Height of the embryo is 1.1 millimeter. 20 hours after fertilization. Somites are 22. Embryonic fin fold on the ventral side extends up to the 10th somite. The lens is now fully formed in the eye. Olfactory placode indicated. Copter's vesicle gets diminished. Blood circulation can be seen over to yolk into the rudimentary heart lying anterior to the yolk sac. The heartbeats Heart beats very rapidly. Some dendritic melanophores appear over the yolk. Next, coming to 22 hours after fertilization. Somites are 24 in number. The embryo encircles the yolk and covers practically the entire capsule. Tail is free. Olfactory pits and auditory vesicles are now well pronounced. Some melanophores appear above the neural cord, over the trunk and caudal regions. Heartbeat is very rapid. Copter's vesicle disappears 24 hours after fertilization. Twitching movement of the embryo is vigorous and lashes the tail vigorously against the capsule, thereby springing up the head, which suppresses the wall and brings the embryo out. The newly hatchling larva is dull brown in color, 2.7 mm long and 1.1 mm height. The fin fold originates dorsally at the second myotome, runs ventrally the region of the vent and goes up to the yolk sac. Third and fourth ventricles of the brain are prominent. Eight dendrite melanophores appear at the caudal region. Linear melanophores appear at the caudal region on the lateral side of the body. Next, 3 hours after hatching. Its length is 3.1 mm and height is 1.4 mm. The heart is two-chambered. Circulation can be seen around the notochord in addition to the brain and yolk. Blood corpuscles are reddish yellow, showing information of hemoglobin. The anal opening is marked by a slight invagination. Next, 8 hours after hatching. Its length is 3.5 mm and height 1.1 mm. Myotome count is 31. Bulge of the yolk is reduced. Some more melanophores appear on the ventral side of the notochord and the dorsal side of the body. Circulation is conspicuous at the optic region. Some pigments are interconnected along the intermyoformal septa. Now look at the picture of this marl fish when they are in larval stage and the adult marl. 15 hours after hatching, its length is 4.2 mm and height is 1 mm. Auditory capsule moves closer to the eye. Pigments over the iris are now denser. Mesenteron is seen as a blind tube and nephridial duct are conspicuous. Location of pectoral fin is marked by a thickened conical patch. 24 hours after hatching. Its length is 4.3 mm and height 10 mm. Myotome count is 32. Buccal imagination appears. Pectoral bud is now well pronounced. The air sac is differentiated as small tube below the pectoral fin bud. Eyes are fully pigmented. 36 hours after hatching. Its length is 4.4 mm. The pectoral fin is a round membranous flap and is being actively used for free movement. Mouth is formed as terminal opening. Vent formed. Rudimentary gill opening and nasal pits differentiated. A thick band of melanophores run from the post-orbital region to base of the pectoral. 2 days after hatching, Its length is 4.6 mm. The pectoral fin is paddle shaped with undulating dorsal margin. Larvae move horizontally in schools. Lower jaw is well developed. Trunk region is a little constricted at the nape. Only a vestige of the yolk is seen. 18 days after hatching, its length is 6.3 mm. Dorsal contour of the head is convex. Basal thickenings appear in the dorsal and ventral fin folds. Light caudal rays can be distinguished. The middle ones showing articulation, the caudal fin is dark along the margin with bright yellow blotch in the middle. Silver and greenish pigments appear on the orbital rim and also on the post orbital region. Now, let us see the changes that happen that looks after 20 days after hatching. Now its length is 7.2 mm. The fry assumes nearly the adult characteristics except the color pattern. Rays starts forming in all the fins from the 18th day onwards. There are 14 rays in caudal, 19 in anal, 25 in dorsal, and 17 in pectoral at this stage. The fry has characteristic iridescent lemon yellow and dull longitudinal blotch bands. Aerial respiration is noticed. The fry move actively in schools. The ventral fins are differentiated on the 23rd day when the fry measures 14 mm. Scales appear on the body by the 26th day when they are 18 mm long. Now let us learn about the life history of cultivable prawns. Generally, the pineid eggs hatch and metamorphose through four distinct stages: Nauplius, zoea, mysis, and post-larval stages. The first one is Peneus indicus. So this species prefers higher salinity. The post-larva range in size from 5 to 10 mm in length. Post-larvae are transparent, white in color, excepting for pink coloration at the tip of the rostrum and the tip of the telson. Antennular peduncle tipped by light reddish coloration. Light yellowish color on eye stalk. A few branched brownish chromatophores tinged with yellow are present on the mid carapace and on few of the mid abdominal segments and telson. A single dorsal tooth is present on the rostrum. The rostrum extends slightly beyond the eye in the first post larvae. A well developed supraorbital hepatic spine is present. A median dorsal and lateral spine is present on the fifth and sixth abdominal segments. Anal spine is also present. Post larvae swim fast, moving along the edges and surface. This is the picture of a prawn. These are the eyes, eye stalk. This is called the anterior portion. It is called as the rostrum. And this is carapace, which covers the thorax, which is called as carapace. And these are the abdominal segments. And this is called as a telson. And these are the uropods. And now the lower legs are called as pleopods. So uropods and pleopods. Pleopods are also called as swimming legs. Then in the anterior region, they have periopods, pleopods and periopods. So periopods are called as walking legs as pleopods are called as swimming legs. Then in the anterior side, this long antenna also we can see. Next, it has two stages in the life cycle, estuarine phase and marine phase. So what is it we can see in marine phase? Eggs, that is nucleus, which changes into zoe. Zoe changes into mysis. Then the post larva, it enters into the estuarine phase. This larva then changes into juvenile, and then the juvenile again it enters into the marine phase to lay the eggs. These are the different larval stages of prawns. Now these are the different colors of prawns. The late mysis measures 3 to 3.5 mm in length. The size range of post larvae is 3.75 to 4 mm. Rostrum is very small, falling short of the middle of the eye stalk. The tip of the rostrum is acutely pointed. Two principal teeth are born at the base of rostrum and one smaller tooth in front of the first principal. A dorsal spine present on sixth abdominal segment only. Post larvae have characteristic brown color, reddish pigmentation on the ventral side of abdomen, present on the entire telson. In later post larvae on the sides of the carapace, bluish brown spots concentrate to form a band. A M-shaped bluish black patch is also seen on the dorsal side of abdomen at this stage. The next one is Metapeneus brevicornis. The size range of the post larvae is 3 to 3.5 mm. Rostrum is short. It just touches the border of carapace. It is brown in color. In juveniles, rostrum shows only slight increase in length, exceeding the tips of the eyes, while the basal elevation is more pronounced. Dorsal teeth are 7 in number. Rostral length is 2.2 mm. Deep yellow coloration is on carapace. Black red bands along the latero-ventral aspect of abdomen. Red spots are seen on the uropods and telson. Let us look at the summary of this chapter. Rohu attains maturity at the end of second year in its life. It spawns only once in a year. Eggs of rohu are round, transparent, non-adhesive, and red in color. Yolk sphere is without oil globule. Newly hatched rohu larva has transparent, laterally compressed body and is characterized by the presence of gill slits, pectoral fin, and median fin fold. Larva of rohu takes 20 to 25 days to get all adult features. The fertilized egg of maril is round, non-adhesive, buoyant, and straw yellow in color. Six somites are seen in the embryo of marl after 14 hours of fertilization. Common exotic carps are silver carp, common carp, and grass carp. In marls, scales appear on the body by 26th day of hatching. Common cultivable prawns are Peneus indicus, Metapeneus monoceros, and Metapeneus breviornis. The larval stage of prawn are Nauplius zoe, mysis, and post-larval stages. Let us see the questions that frequently that are asked. Write about life history of rohu. Write about life history of other cultivable fishes. Write about life history of marl. Write about life history of cultivable prawns. Then the questions that are asked to write in 10 lines. Write about Peneus indicus. Write about Metapeneus monoceros. Give a brief description about Metapeneus brevicornis. Write in detail about silver carp, grass carp, and common carp. So with this, we have come to the end of the chapter. Hope you all have understood. Thank you.